Hello everyone. In this session we are going to discuss about nucleic acids. So which comes under the paper 5 cell biology and biotechnology. So mainly in this subtopic, that is nucleic acids, it covers about the introduction, identification of genetic material by different experiments performed by Griffith, Avery, MacLeod and McCarthy, Hershey and Chase experiments. And later you need to study about the chemistry of nucleic acids, structure of DNA, Watson and Crick model, that is DNA double helix model, mechanism of DNA replication, and the experiments conducted by Stahls. And there are different forms of DNA, that is A, B, Z, again B1, B2, and DNA repair, photoreactivation, recombinational repairs, and types of RNA, that is again in that you need to study about transcription and translation. So this is how the nucleic acid subunit, these are the subtopics which comes under the nucleic acid. So before knowing about identification of genetic material, first we need to know what do you mean by nucleic acids and how the structure of DNA is, and later we'll move on to the experiments. So in this session mainly we'll concentrate on what are nucleic acids and what are the composition, double helix model, and what are the functions and properties of the DNA. So let's begin with our session, that is nucleic acids. So we all know that the nucleic acid, they are nothing but the macromolecules with acid properties. That is why the name comes the acid. And it was first isolated from the nucleus of the cell, that is the pus cells, and hence named it as nucleic acids. So usually it consists of the carbon, hydrogen, oxygen and nitrogen along with the phosphorus. The nucleic acid was first isolated in 1868 by Friedrich Mischer from the nuclei of the pus cells. So as he was observing the pus cells or the pus which was in the bandage of the wounded man, and he found out there are some small cells like structure which he named it as the nucleon. And later coming the Altman, he gave the name the nucleic acid in the year 1889. But the nucleic acids are found in all the organisms like plants, animals, microorganisms, but they are found in the nucleus as well as in cytoplasm. So these are the two important pioneers who gave the terminology and who found the Friedrich Mischer and the Richard Altman. So next we'll see the chemistry of the nucleic acid, that is mainly we'll see about the DNA. So this double helix model was proposed by Watson and Crick. So this nucleic acid molecule, they are nothing but the long chain of the polymer, and they are composed of, as we have discussed, that is monomeric units called as nucleotides. So here they are composed or they consist of nucleoside and the phosphate group. So that is why due to the presence of phosphate, then only the acidic nature comes, then only we'll call them as the nucleic acid. But also along with that it contains nucleoside containing pentose sugar and the nitrogenous bases. So these bases and the sugar, they make a composition or they will have the double helix model so that they bond to each other and they are strong enough. So the sugar is which consists in RNA, that is ribose sugar, but whereas in DNA it will be the deoxyribose sugar. So this is how the structure of DNA which was proposed by Watson and Crick looks like. So Watson and Crick in the year 1953 they designed the structure of DNA and they gave their name Watson and Crick model, and they also received the Nobel Prize in the year 1962. So according to Watson and Crick, the DNA is in the form of double helix, but this is what we are knowing. But there is lot of work which was done by the Rosalind Franklin, which you can see in the lower part of this picture. The female who gave the X-ray diffraction method. She was the pioneer, or she was the main person to come to know the DNA structure, because she made the X-ray crystallographic images which was given by the Watson and Crick. So that model which was proposed by Watson and Crick, the images were given by the Rosalind Franklin. So she didn't get any Nobel Prize, but we are having different types of institutes under her name for her the contribution of this double helix model. So let's see how the deoxyribose nucleic acid, what are the composition. So this DNA, it is nothing but the nucleic acid, and it is macromolecule, and this macromolecule, it is made up of two chain, that is each chain it has having polynucleotide chain, and for that we will call it as nucleotides. The nucleotide, they are made up of three important chemical components, that is phosphoric acid, deoxyribose sugar, and nitrogenous base. So if you are telling about the ribose sugar, then it will be a RNA, that is ribonucleic acid. So the nitrogenous bases are nothing but adenine, guanine, thymine, and cytosine. But remember, the cytosine will be replaced by the uracil or the uracil. So there what happens, the RNA will be composed. So now this is how the four important nucleotides are there. So these four different types of nucleotides, they are having the terminologies like AMP, that is adenosine monophosphate, or also it is also called as adenylate acid. GMP, that is guanosine monophosphate, guanylic acid. TMP, that is thymidine monophosphate, thymidinic acid. CMP, cytidine monophosphate, it is also called as cytidinic acid. So these are the important four different types of nucleotides which are consisting in the DNA. So this is how the chemical structure of DNA looks like. So as you can see in the first portion, the sugar phosphate, which is the backbone, the sugar phosphate, the backbone, usually how the structure or how it is composed of. And these are the four important types, that is adenine, guanine, thymine, and the cytosine. So this is how the double helix model looks like, because the helical formation, it takes place in this way, and it has got different types of bonding, as you can see. So that makes the DNA a stable molecule. So this is how the DNA double helix model looks like, and as you can see, the DNA, when it is transferred to RNA and RNA to protein, this is called as central dogma of molecular biology. So once the DNA to DNA, so if you are having the amount of DNA, if you want to produce more amount of DNA now, so that more amount of DNA has to be amplified. So that is called as replication. So if the DNA has to be changed to RNA, then it will be undergoing transcription, and RNA has to be done to protein or the amino acid, then it is called as translation. So these are the important steps that comes under the DNA to protein, like or DNA to DNA, DNA to RNA, and RNA to protein. So next we'll see this Watson and Crick model, as they have proposed the double helix model. So in each nucleoside they had the pentose sugar which is attached to the nitrogen atom, and this nitrogenous base, it is linked by the glycosidic linkage. We call it as N-glycosidic linkage also. And the phosphoric acid, the molecule is linked with the sugar of nucleoside. So as you can see in the picture, how the nitrogenous bases are attached. So there is a phosphate backbone, and that phosphate backbone is having the link between the DNA molecule, and hence the DNA looks like the double helix model. And many nucleotides are linked together to form nucleotide chain. So nucleotides are nothing but the nucleotides one upon the another, like A, T, G, C. They combine together, and they are combined, or the two nucleotides are joined by the phosphodiester linkage, and this is formed between sugar and the nucleotide. So as I was telling, the DNA molecules is having two polynucleotide chain, and these polynucleotide chains, they are linked. For example, adenine is linked with the thymine, AT. Then we are having the similarly with guanine is linked with the cytosine, so GC. So as it tells that A is linked with T and G is linked with C. So as I already said, they are linked by the hydrogen bonds also. So AT, they are linked by 2 hydrogen bonds, whereas G and C, they are linked by 3 hydrogen bonds. So this is how the polynucleotide chains are formed, by linking hydrogen bonds also. So this is the structure which I already shown. As you can see, the thymine, adenine, cytosine, guanine, and also the deoxyribose sugar, how they are linked with the phosphate bond and the hydrogen bond. So this is how the linkage of the molecule, if you are studying the chemical structure, how they look like. Pentose sugar is there. This is why the pentose sugar, it has got 5-sided, and the nitrogenous bases along with the adenine and guanine, even that is also having the pentose lining. Along with that, the nitrogen bonding is present, and the phosphate or the diester linkage, it is also present. So the same structure. Okay, this is the exact structure which I can show, how the structure, like this is the ribose sugar as the pentose sugar, and the base you can take it as adenine, guanine or anything, and phosphate linkage, which gives the bonding between N-glycosidic linkage, and the hydrogen bondings is also present. So along with this, we'll move to the one more important concept, that is Chargaff equivalent rule. So it is nothing but the number of adenine, guanine, or you can tell number of adenine is equivalent to thymine, and number of guanine is equivalent to cytosine. It is nothing but the amount of A is equal to amount of T, and the amount of G is equivalent to C. So this is how he gave the Chargaff equivalence rule. The two chains of DNA are complementary to each other. So number of nucleotide present in one chain has to match with the number of nucleotides with the another chain. So that is called as Chargaff equivalence rule. So he is the person, the Irwin Chargaff, who gave, that is A is to T is equal to G is to C. So that is called as in the ratio of 1:1. So as you know, the DNA is the double helix model. They run anti-parallel to each other, and that is how they form spiral staircase like structure, and this DNA molecule is in the form of double helix. These polynucleotides, they are coiled together and they form the double helix model. And along with that, the diameter, this is very important, that is having 20 Armstrong, and along with that, the DNA helix, it is having two important external grooves, that is major groove and minor groove, or it is also having the major groove will be deep, and whereas minor groove, they are shallow and narrow. The distance between the two nucleotide, this is also very important, 3.4 Armstrong, and this double helix model, which was given by Watson and Crick, it is also called as right-handed helix or B-DNA, B type of DNA. And there are different types, that is right-handed DNA is also present, and that is left-handed DNA, that is will be the A-DNA. So this type of model, which was given by Watson and Crick model, so that is right-handed helix, and that will be a B type model. So what are the properties of DNA? So as we know, the size of the DNA molecule. So each animal or each species, our DNA to DNA in organism, it will be changing. Species to species, organism to organism, the DNA will be variable. And fragility of the DNA molecule. So we know the fragility, the DNA is very much stable, and sometimes, for example, when you are making experiments in your molecular labs, like when you are pipetting out the DNA, when you are pouring the DNA from one vial to another vial, so that time breakage of DNA can takes place. So that is how so much very fragile they are. Denaturation, you already know the DNA, if you are heating, they get denatured. and at certain temperature they get denatured. So they are kept in very much low temperature in the different types of cellular and molecular labs. Renaturation is nothing but once the denatured DNA can be renatured by giving the optimum temperature or bringing back to the neutral condition, so that is neutralizing or cooling effect. And effect of pH on DNA, it is nothing but there what happens, the pH has to be denaturing occurs at around 11.3 in alkaline condition, so the stability has to be maintained. And along with that, the hydrogen bonding and hydrophobic bondings are very important. And hyperchromic effect, so it is nothing but the light has to pass, that is how we already studied the crystallography method. So in crystallography what happens, once you light monochromatic light has to be passed, then only the structure of DNA you have seen. But here the DNA molecule, it absorbs the light energy, and this property is individual on the basis, maybe on adenine, guanine, cytosine and thymine. And this intact DNA, it absorbs less light energy, and it is based on the packed in the double stranded. So these are the important properties of DNA. So we'll see the functions of DNA. So as we know, the DNA plays very important role in our heredity, because this DNA has to be transferred from one generation to the another generation, like parents to offsprings. And this DNA is very important, that is, it carries the genetic information, and along with that the DNA controls all the development processes, and DNA synthesizes the RNA. So as we have already discussed about the central dogma of molecular biology, where DNA is changed to RNA, RNA is changed to amino acids by different types of processes like replication, transcription and translation. And this DNA has the genetic information for the protein synthesis also. So we have studied in this session mainly about the nucleic acids, under that Watson and Crick model, nucleosides and nucleotides of DNA, and in that we have studied how the structure, chemical composition by nitrogenous bases and the bondings, N-glycosidic linkage, hydrogen bondings. So in this topic mainly the Watson and Crick model can be asked for 7 marks, and the properties of DNA and functions of DNA can be asked for 3 or 4 marks also. So in this session we have completed the very important concept about the nucleic acids. So that's all about this session. Thank you.